So first of all, a very good evening to all of you. So today basically I will be going to deal with the chapter sexual reproduction in flowering plants. Now, as you all know that it is one of the most important chapters for REIT exam, and around 4 to 5 questions are definitely going to come from this chapter, and is also one of the lengthiest chapter in biology. So I had decided to divide this chapter into two parts, part 1 and the part 2. So basically in this video I will be going to discuss the part 1 of this chapter, the sexual reproduction in flowering plants. Now in this part I will be going to discuss the structure of a flower, the male and the female reproductive parts of a flower, the development of the male spore and the male gametophyte, whereas the development of the female spore and the female gametophyte. So let us move into the chapter. So basically, as you all know that the all flowering plants show sexual reproduction. So basically, the thing which actually separates a flowering plant from a sexually reproducing plant from a non-sexual reproducing plant is the flower. So basically, the flowers are the site for sexual reproduction. Now, as you all know that the flowers basically contains four holes: the calyx, corolla, androecium, and the gynoecium. The calyx and corolla are called the accessory holes or the non-essential holes of a flower, whereas the androecium and the gynoecium are called the reproductive holes or the essential holes of the flower. And all these four holes are arranged on the receptacle called the thalamus. Now, it is also known to you that the flower is also called a modified suit or a modified condensed suit because the nodes and internodes are present in flower, but those are present in a condensed state. And also the presence of these floral leaves, the calyx and corolla. That is why the flower is called a modified suit. Now coming to the non-essential floral organs. So as I already told you that these non-essential floral organs are the organs or the part of a flower that basically does not take part in reproduction. So basically there are two actually holes which are non-essential. Those are calyx and the corolla. So now coming to the first non-essential floral holes, that is the calyx. Now I already had discussed almost all this in morphology. I will be just giving you an overview of this. So as you all know that the unit of a calyx is called the sepal, and it is green in color mostly. So basically the role of the sepal is to protect the flower during the bud condition, and also in certain condition it helps in photosynthesis. And there is another essential hole called the corolla. The unit of corolla is called the petal, and as you all know that the petals are brightly colored. So basically the main function of the petal is to attract insects or the pollinators for pollination. And also the stalk of a here mistakenly it is written flower, it must be a actually inflorescence. So the stalk of inflorescence is called the peduncle. Now there is another thing called the perianth. In certain flowers you won't be able to distinguish this the corolla and calyx into separate units. Those flowers are called the perianth, as for example in case of tuberose or polyanthus. Now coming to the essential floral holes. Now as you all know that there are two essential holes of a flower, the androecium and the gynoecium. So coming to the first essential hole, that is the androecium. Now the units of androecium is called the stamen, and as you all know that the androecium is the male reproductive part of a flower. And the stamens are also called microsporophylls. One thing that I want you all to remember is that the term micro is always associated with the male reproductive system. Be it plant or animal, always the term micro is associated with the male reproductive system, whereas the term mega is associated with the female reproductive system. So keep this in mind. So basically the sporophyle signifies the organ or the part where spores have been present. So as you all know that the spores of a flowering plant are the pollen. So basically the pollens are present in this stamen, that is why these are called microsporophyll. Micro because it is the male part. Now, as you all know that there are two parts of a stamen, the filament and the anther. The anthers are basically two lobes, that is why it is called diathecus. Whereas in case of Malvaceae family, it is one lobed and it is called monothecous condition. Whereas in China rose, the anther lobe is kidney shaped and it is called reniform. Now, another thing is that a single anther lobe basically contains two pollen sacs. You can see here the cross section of an anther lobe. This is a single anther lobe and it contains two pollen sacs. So basically, two anther lobe will contain four pollen sacs. So that is why each anther lobe is called a tetrasporangiate because of the presence of four pollen sacs. Four means tetra, that is why tetrasporangiate. And inside this pollen sac is present the pollen grain. Now coming to the female reproductive part, that is the gynoecium, and the unit of gynoecium is are the carpels or the pistils. Now the gynoeciums are called the megasporophyll. As I already told you, sporophyll because it bears the female spore, that is the egg, and mega because it is a female part, that is why megasporophyll. As you all know that the gynoecium, that each carpel consists of three parts: stigma, style, and ovary. The stigma is the part where the pollen actually falls after pollination. The style is the elongated part of the stigma from where the pollen tube passes to the ovary, and the ovary is the bulged out portion of the carpels. Inside the inner part of the ovary is called the locule, and within the locule there is a certain cushion-like tissues called the placenta, and within this placenta are present the ovules. So ovules are present in the ovary. Now there are different types of gynoecium like monocarpellary, as you all know, mono means single. So basically when only one carpel is present, it is called monocarpellary. Multicarpellary, when more than one carpel is present. Apocarpous, if the carpels are free, and if the carpels are fused, then it is called syncarpous. Now these things I have already discussed in the chapter morphology in detail. So you guys don't have to read this from here. So one thing that I want you all to remember is that the number of ovule in a ovary can be one to many. Like the ovules can be one in case of wheat, paddy and mango, whereas the ovules can be many, for example in papaya, watermelon and orchids. I want you all to remember these two examples because these two are a very important question for the NEET. Now let us look deep into the anther lobes. So that is the pollen sac or the microsporangium. Sporangium is basically the site where the microspores are present, that is the pollens are present, and as you all know that is the pollen sac. So let us move into the structure of a pollen sac or a microsporangium. So if we see the TS, this is the TS or the transverse section of a anther lobe. So we will see that the pollen sac basically appears circular in appearance and it is covered by different layers. So if we look into the layers covering the pollen sac, we will find four different layers. The outer layer is called the epidermis, then the endothecium, then the middle layers, and then the tapetum. So let us look into each of these layers in detail. So first of all, coming to the epidermis. So basically epidermis is the outer single layer of the anther and it is protective in nature and it is a single celled layer. Now coming to the endothecium. Now endothecium is present below the epidermis. Now endothecium is also a single layer, but one thing is that basically the layers of endothecium becomes thickened by the deposition of certain complex carbohydrates such as alpha cellulosic fibers. Now remember one thing that these are the radial walls. The side walls are called the radial walls of the endothecium, whereas this is the inner wall of the endothecium and this is the outer wall. So basically the deposition of alpha cellulosic fiber takes place in this radial and this inner wall, whereas the outer walls remains thin. So this actually has a role to play during dehiscence of anther. I will come to that later. So basically the main thing that you have to remember is the deposition of alpha cellulosic fiber in the radial and inner wall of the endothecium, and these things is basically hygroscopic in nature. So this has something to do with the dehiscence of anther. Now coming to the middle layer. Now middle layer is basically consists of parenchymatous cell. So and this layer is 1 to 3 celled layer. Means as you see that epidermis and endothecium are single wall layer, whereas the middle layer is 1 to 3 layers normally, but the number of layer can vary, and the function of middle layer is to store food. in these parenchymatous cells. And now coming to the innermost layer of the anther lobe, or the layer surrounding the pollen sac, that is called the tapetum. Now tapetum is a nutritional layer because it provides nutrition to the growing pollen grains, and it is polyploid in nature due to endomitosis. Again, endomitosis is something is special type of cell division in which the chromosome divides but the cell actually does not divide. So basically what happens is that a single cell contains more than normal number of chromosomes. That is, the ploidy number basically in the tapetum cells are triploid or tetraploid, means 3n or 4n, whereas we know that the normal number is diploid, 2n. So the polyploid nature is found in the tapetum. It is very important. And another thing is that the cells of the tapetum secrete certain hormones. And another important thing, that is the Callase enzyme. Remember the term Callase enzyme. We will come to the function of this later. Now there are two types of tapetum. One is the amoeboid tapetum and the another one is the secretory tapetum. Now the function of tapetum is to absorb the food from the middle layer and to provide nutrition to the microspores or the pollen grains. And as I already told you, it is of two types, amoeboid and secretory. Now coming to the amoeboid tapetum. Now this type of tapetum layer is normally found in the primitive angiosperms and not in the advanced angiosperms like Typha, Elisma, Tradescantia, etc. And the, what this type of tapetum does is that this tapetum layer absorbs food from the middle layers. So the middle layer immediately degenerates because it absorbs whole amount of food from the middle layer. Now in the beginning, the tapetum cell stores food materials in the tapetum layer. Now the tapetum cells convert the absorbed food into a certain bodies called the protoplast bodies. Now this innermost layer of tapetum now dissolve and resolve these bodies into the cavities of the microsporangia. So now inside the pollen sac, these protoplast bodies are called the periplasmodium. So it basically provides nourishment to the growing microspore. So what happens is that in this type of tapetum layer, the middle layer degenerates and the tapetum layer also degenerates to form the periplasmodium bodies, which actually provide nourishment to the microspores. Now coming to the glandular or the secretory tapetum. Now in this type of tapetum, or the cells of these tapetum layers remains intact throughout the microspore development. Now it absorbs nutrient from the middle layer and secrete into the cavity of the microsporangium, that is the pollen sac, and does not store it, and it does not absorbs the whole amount of food from the middle layer, as a result of which the middle layer cells also remains intact. Now in this type of the tapetum, the cells basically forms special granules called the pro-pubis body, within which the food is stored. And now this pro-pubis body get covered by a substance called sporopollenin, and it is now called the pubis body. Now what is sporopollenin? I will come to that later. Now these pubis bodies are then released into the pollen sacs, and they helps in the nourishment of the pollen grains. And the sporopollenin, which forms the wall of the pro-pubis body, actually helps in the forming the outer layer of the pollen grain. So as you can see in this diagram, this is the amoeboid or the primitive type of tapetum. In this, the tapetum layers actually degenerates, and these are the protoplasmic bodies basically surrounding the mature pollen, and these are the protoplasmic bodies, and the pollen grains actually gains nutrient from these protoplasmic bodies. Whereas as you can see in the secretory tapetum, the tapetum layers remains intact. Sorry. Now coming to the microsporogenesis. Sporogenesis basically signifies the formation of spore. Genesis, the word has come from synthesis, and spore synthesis. So basically, and micro is the formation of male spore. So basically the formation of the microspore or the pollen is called the microsporogenesis. Now at the initial stage, the anther is a mass of undifferentiated meristematic cells, which is surrounded by just a epidermis layer. The four cells located just below the epidermis becomes enlarged, and these cells are called the archisporial cells. This is the archisporial cell. So basically during the development of anther, the anther are undifferentiated, the cells of the anthers are undifferentiated and only is surrounded by epidermis. Some cells become enlarged to form the archisporial cells. Now the archisporial cells divides to form the upper primary parietal cell and the inner primary sporogenous cells. Now primary parietal cell undergo further division to form a series of 3 to 5 cells layers making the wall of the anther, that is the endothecium, middle layer and the tapetum. So these layers are formed from the division of the primary parietal cells. Now the primary sporogenous cells basically divide by mitotic division to form sporogenous cells, and later it differentiates to form the microspore mother cell. Now microspore mother cell undergo meiosis, you can see here. So basically as primary sporogenous cells undergo mitotic division, so the ploidy of primary sporogenous cell was 2n and the microspore mother cell also remains 2n. Now this microspore mother cell or the pollen mother cell undergo meiosis division to form the microspore tetrad. Now as meiosis division is occurring, so the microspore tetrads are basically haploid in number. Now at this stage, all the 4 microspores are attached together with the help of a carbohydrate called the callose. These are the callose repollination, which actually joins these 4 microspores. Now, but as we all know that during pollination, a single pollen grain is the pollen grains are not actually removed in the form of a tetrad; they are singly released. So this dissolvement of the callose is done by the callase enzyme, which is secreted from the tapetum layer. I told you that I will say that function of callase enzyme later on. So the function of the callase enzyme is to dissolve this callose layer between the microspore tetrad to form single microspores. Now in dicot, these microspores are arranged one upon the other, and this condition is called the tetrahedral tetrad. Whereas in case of monocot, these microspores are arranged laterally, and these are called the isobilateral tetrads. So in this way, the pollen grains are formed or the microspores are formed. So this is called microsporogenesis. Now coming to the structure of the pollen grain or the microspore. Now pollen grain represent the male gametophyte. It is usually 25 to 30 micrometer in diameter, and there are the pollen grain is basically covered by two walled layer. The outer wall is called the exine and the inner wall is called the intine. Now exine is made up of sporopollenin. Now sporopollenin, I told you that the substance which actually forms the wall of the obese body, the sporopollenin, it helps in the formation of outer wall of exine, and it is the most highly resistant organic substance known on the earth. So basically this sporopollenin makes the pollen grain a fossil protective substance. Means we saw that during fossils formation the pollen grains are found as fossils. So what makes this pollen grain such strong to bear such high pressure and thrive through these years? So this is the sporopollenin, which makes the pollen grain a fossil protective substance. And there are certain places in the outer wall where the exine layer is either absent or very less present. So those layers are called the germ pore. Now, and the inner wall is called the intine. The intine is basically made up of pectocellulose, and this intine layer basically comes out of the germ pores during germination. Now, in insect pollinated plant, the exine has certain thing which is called the pollen kit. Now, pollen kit is basically yellowish and a viscous, sticky and oily layer, and it is made up of lipid and carotenoids. So the function of the pollen kit is so that when the insect actually comes for gathering the nectar, this pollen kit which is present on the wall of the pollen actually helps in attaching the pollen to the hairs or to the body hairs of the insect so that the pollination can occur. So pollen kit is found in the pollen grains of the insect pollinated pollens. And now when the pollen grain is mature, it contains two cells, the vegetative cells and the vegetative cell and the generative cell. Now the vegetative cell is bigger and has abundant food reserve, whereas the generative cell is small. So in this, when over 60% of angiosperms, these pollen grains are basically set in these two cell stages, that is containing the vegetative cell and the generative cells. Whereas in the remaining species, the generative cells normally divides mitotically to give rise to two male gametes, and the pollen grains are set in this three cell condition, that is the vegetative cell and the two male gametes. But in most of the angiosperms, the setting of pollen grains takes place in two cell condition, that is pollen grain containing the vegetative cell and the generative cell. Now I will come to the formation of vegetative cell and generative cell later on. So now coming to the development of male gametophyte, that is the male gamete. Now this process is also known as microgametogenesis. Gametogenesis means formation of gamete and micro means male gamete. So basically germination of pollen grains, that is formation of the generative and vegetative cell, takes place before the dehiscence of anther. And as it takes place before dehiscence of anther, so it is called the precocious germination. Now we can divide the whole process or the whole process of germination of pollen grains into two parts. One is the pre-pollination development and the another one is the post-pollination development. Now coming to the pre-pollination development. So nucleus of pollen actually divides by unequal mitotic division. You can see that the nucleus of the pollen divides into two of unequal size. One is the bigger and the another one is the smaller nucleus. So the bigger nucleus actually forms the vegetative nucleus or the tube nucleus, whereas the smaller nucleus is called the generative nucleus. Now following cytokinesis, large cell with larger nucleus is called the vegetative cell, whereas the smaller cell with the small nucleus is called the generative cells. Now, I already told you that in 60% of angiosperms, the pollen grain is set in this stage, that is containing the vegetative cell and the generative cell. Now, pollen grains form on the stigma, so and absorbs moisture and sugar from stigma. So, due to this, the volume of the internal content of cytoplasm of the pollen grains increases. As a result of which, it exerts pressure on the intine layer. As a result of which, the intine actually comes out of the pollen grain through the germ pore in the form of pollen tube. So firstly the vegetative cell enters the pollen tube. Now inside the pollen tube the generative cells now divide mitotically to form two male gametes. So this three cell stage represent the mature male gametophyte. So and this happens after the pollination. So in this way the male gametes are formed. And this male, the tube nucleus then enters the embryo sac and the male gamete then fuses with the egg to actually result in fertilization. We will come to that later on. So now we saw that how the pollen grains are formed, what is the structure of pollen grain, and how the male gametophyte is formed, the male gamete is formed. So now the main thing is the anther must be liberated so that pollination occurs. So this is the dehiscence of anther. So during the maturation of anther, various changes take place in the wall of anther. Now in the beginning, the middle layer degenerates due to absorption of food by the tapetum. When microspores are formed in the pollen sacs, Ubi's body are formed in tapetum and then the tapetum degenerates. So in this way, a mature anther basically has two layers only, an epidermis and a endothecium, because the tapetum and the middle layer degenerates. Now normally dehiscence of anther takes place during dry season. Okay, so in dry season, endothecium lose water because of the hygroscopic nature of the alpha cellulosic fiber. Now wall of the endothecium cells try to contract due to the loss of water, but inner and the radial wall do not contract due to the presence of this thickening of alpha cellulosic fiber. I already told you. So what happens is that this creates a tension over the entire surface of anther, and due to this tension, the basically anther walls breaks out from the stromal area. These areas are called the stromal area, where actually the deposition of alpha cellulosic fiber is less. So and the dehiscence of anther takes place and the pollen grains are released. So in this way the dehiscence of anthers takes place. Now coming to pollen allergy. Now pollen grain of many species causes severe allergy in some people, leading to chronic respiratory diseases like asthma, bronchitis, etc. This all you know. Some sources of pollen allergy are carrot grass, chenopodium, ricina, sorghum, etc. The one thing I want you all to remember is that The Parthenium or the carrot grass basically is not an indigenous product of India. It came to India as a contaminant with imported wheat, and now due to its high rate of reproduction, it is found almost everywhere, and it causes pollen allergy. So the pollen does not only causes trouble to human beings. These are of social importance, or it is of importance to mankind also. So the uses of pollen grains are, the pollen grains are a very rich sources of nutrient. The pollen tablets are used as food supplements in many Western countries, and pollen consumption has been claimed to increase the performance of athletes and horses. So these are the certain uses of pollen grains. Now it is possible to store pollen grain of a large number of species for years in liquid nitrogen, that is minus 196 degrees Celsius. Now, the viability period of pollen grain, that is the functional period of pollen grain, is highly variable. Like in some cereals like rice, wheat, the pollen grains lose viability within 30 minutes of falling on the stigma, whereas in some members of Rosaceae, Leguminosae, and Solanaceae, they remain viable for months. Now, in Calotropis and Orchidaceae family, that is orchids, the pollen grains join together to form pollinium. This is the pollinium structure, and basically this the dispersal of pollen grains does not take place singly, but it occurs as a pollinium, and one pollinium is dispersed as a whole. Similarly, in Cuscuta, more than 4 pollen grains are found in tetrad, and this condition is called polyspory. I already told you that a tetrad contains 4 pollen grains, but in case of Cuscuta, more than 4 pollen grains are found in the tetrad, and that is called polyspory. Now, the pollinium of Calotropis is also called the translator apparatus. Remember the name. And the pollen grain of Ambrosia causes hay fever. So, with this, I came to the end of the male reproductive system of plant and the process involved with it. So now we know that how the pollen is formed, how does the pollen look like, how the male gamete is being formed, and what happens after pollination. So now, to complete fertilization, we must also know about the female reproductive system. So let us move to the female reproductive system. So let us look into the megasporangium, that is the ovule. Why mega? I already told you, because it is the female reproductive part, and sporangium because it bears the female spore, that is the egg. So ovule is called the megasporangium. So now, as I already told you, that the cavity inside the ovary is called the locule, and within this locule there is some cushion like tissues called the placenta, and within this placenta these ovules are attached, and the ovules can be one to many in different other flowers. Now the ovule is attached to the placenta by the help of a stalk called the funicle. Now hilum basically represent the junction between the ovule and the funicle, and each ovule has one or two protective envelopes called the integuments. The inner integument is called the inner layer is called the inner integument, whereas the outer layer is called the outer integument. Whereas in certain other plants, like in case of lychee, third integument is also present, which is called aril. And it is interesting to note that the edible part of lychee is the third integument, that is the aril. Now the integument encircle the whole of the ovule except a certain region, that is called the micropyle. The region where integument is absent, that is called the micropyle, and the end opposite to micropyle is called the chalaza. Now enclosed within the integument is the whole mass of nucellus. Now inside this whole mass of cells is called the nucellus, and within the nucellus is located the embryo sac or the female gametophyte, because it contains the egg. So now in some angiosperm, the entire nucellus is utilized to form the embryo sac, whereas in some, a part of the nucellus remains within the seed, and that is called the perisperm, as in black pepper and beet. Remember the example, this is very important for a NEET exam, that what is perisperm? The perisperm is the remains of nucellus that remain after forming the embryo sac, and it occurs in black pepper and beet. Now the funicle is attached to the main body, and it forms a ridge that is called the raphe. So now this is the overall structure of a ovule, and we will come to the structure of embryo sac later on. So now coming to the type of ovule. So on the basis of integument, there are basically three types of ovule. Now, unitechnic ovule, the name suggests that when the ovule contains one integument, as in case of gymnosperms, whereas there is a bitechnic ovule, bi means two, that is the ovule is surrounded by two envelope, and as it occurs in angiosperms, whereas there is certain ovule where there is no integument, and that is called the atechnic ovule. Example in case of Loranthus. Now, on the basis of configuration or the shape of the ovule, the ovules are of 6 different types. So, those are orthotropous, anatropous, hemitropous, campylotropous, amphilotropous, and cercinotropous. So, now let us look into each of these in details. So, now if you look at the orthotropous ovule, this is the orthotropous ovule. It is also called atropous ovule. In this case, the micropyle, the chalaza, and the funicle are in a straight line. As you can see, the micropyle, the chalaza, and the funicle is in a straight line. So this ovule is also called the upright ovule or the erect ovule. The second is the anatropous ovule. Now, if you actually turn the orthotropous ovule at 180-degree angle, then it will form the inverted ovule or the anatropous ovule. So in 80% of angiosperm family, the ovule type is anatropous, and the ovule which actually I already showed in the previous slide was also anatropous ovule. And you will see in most of the explanation of ovule, people use this anatropous ovule. So it is also called the typical ovule. Now coming to the, there is, if you actually turn the orthotropous ovule 360-degree, it will form the cercinotropous ovule, in which the funicle actually becomes coiled around the ovule, and this is found in the opuntia. Now coming to the hemitropous ovule. Now if you actually turn this orthotropous ovule at an angle of 90-degree, you can see this is 0-degree and this is the 90-degree angle. With respect to orthotropous, hemitropous is 90-degree. So basically what happens is that the micropyle and the chalaza are not in a straight line. So this type of ovule is called the hemitropous ovule. Now coming to the campylotropous ovule. Now in campylotropous ovule, the bending or the ovule bends in such a way that the micropyle and chalaza are not in a straight line. That is, it is not 180-degree bend or 90-degree bend. The bending angle is something between 90 to 180-degree, and that is called the campylotropous ovule. Then coming to the amphitropous ovule. Now amphitropous ovule is the ovule in which the ovule becomes curved in such a way that it took the shape of a horseshoe, and this is known as the horseshoe shaped ovule. So this is a very important question for NEET, that what is a horseshoe shaped ovule. So this is the amphitropous type of ovule is called the horseshoe shaped ovule. So these are the different types of ovule that can be found in the flowering plants. Now coming to the megasporogenesis, that is the formation of female spore or the female spore. Like now the process of formation of megaspore from megaspore mother cell by meiotic division is called the megasporogenesis. So what happens is that during the development of ovule, in the beginning, the nucellus consists of undifferentiated cell. There is no embryo sac till now, and the single cell, a random single cell increases in size, and it forms the archisporial cell. This is the primary archisporial cell. Now this archisporial cell divides to form a outer primary parietal cell and the inner primary sporogenous cell. Now this primary sporogenous cells actually directly act as the megaspore mother cell. Now this megaspore mother cells now divides meiotically to form 4 haploid megaspores. Now of these 4 haploid megaspore, one of the 3 of the megaspore degenerates, and one of the megaspore actually gains nutrition from the nucellus and enlarges in size to form the functional megaspore. Now this functional megaspore basically forms the embryo sac. So these are the different stages of megasporogenesis. Now coming to the development of female gametophyte or the embryo sac. Now I already told you that of the 4 megaspore formed, 3 degenerates and one of the megaspores enlarges to form the functional megaspore. Now megaspore is the first cell of the female gametophyte. So the megaspore grows in size by absorbing nutrition from the, this is the case, the megaspore actually grows in size by absorbing nutrition from the nucellus. The nucleus within the megaspore now divides into two, and they move to the opposite poles.
Now the nucleus in each of these pole now again divides to form basically 2 nucleus at each pole. Now these 2 nucleus at each pole now again divides to form 4 nucleus at each pole. Of these 4 nucleus, 1 nucleus from each poles, means 1 nucleus from here and 1 nucleus from here, will move to the center of this megaspore to form the, and they are called the polar nuclei because they came from the poles. Now remaining 3 nuclei in each of these poles are surrounded by cytoplasm, and this 3 cell from the chala toward the chalaza is called the micropyle. Sorry, toward the micropyle is called the egg apparatus, whereas the 3 tail cells toward the chalaza forms the antipodal cell. And both the polar nuclei are present in the central cell. They actually fuse to form a central cell, but the nucleus remains in the form of 2 nucleus, a single cell but 2 nucleus. But just before fertilization, they unite to form the secondary nucleus. So that is the diploid condition, that is 2n. So basically before fertilization, the 7-celled 8 nucleus structure is formed, and this structure is called the embryo sac, because 3 nucleus in this pole get surrounded by cytoplasm to form 3 cell, 3 nucleus in this pole surrounded by cytoplasm to form 3 cell, that is 6 cell and containing 6 nucleus. But the polar nuclei, actually the nucleus does not fuse before fertilization, but they are covered by cytoplasm. So what happens is that this is a single cell, this 3 and this 3, there are 7 cell, but 3 nucleus and 2 nucleus, that is 8 nucleus. So before fertilization, this 7 celled 8 nucleus stays prevails, and this is the embryo sac. But just before fertilization, these 2 nucleus fuses to form a 2n structure, that is the diploid structure. So now this type of embryo sac is known as the polygonum type of embryo sac because it was firstly discovered by Strasburger in Polygonum. Now at this type of embryo sac development, as the embryo sac is developed from a single megaspore, so this type of embryo sac is called monosporic embryo sac development. Now coming to the embryo sac. Now, as I already told you that the 3 cells towards the micropyle forms the egg apparatus. This is the egg apparatus, whereas the 3 cells toward the chalaza forms the antipodal cell. The egg apparatus basically contains a single egg, which is haploid, and 2 other cells, 2 synergids surrounding the egg, which are also haploid. Now the synergids contain some finger-like projections which are called the filiform apparatus. So the function of this filiform apparatus is to guide the pollen tube towards the egg, and the antipodal cells remains toward the chalaza. And as I already told you that the polar nucleus basically get covered by a single cell and they form the central cell. So this is the structure of a basic embryo sac. So now this is the thing basically. So we actually discussed the male reproductive part of a flower, the formation of the male gametophyte and the formation of the male gamete, and we also discussed the formation of the female gametophyte, the formation of the female gametophyte. We discussed the female reproductive system of a flower and also the development of the female gamete, that is the egg. So now if I ask you a simple question, that is, what are the ploidy of these cells? So I want you all to uh, keep this slide here and but pause this slide here and try to answer this for the ploidy of 4 cells individually without knowing the answer, and then resume the video to know the answer of this cell. So basically the nucellus is diploid in structure, the microspore mother cell is again diploid, the functional megaspore is haploid, and the female gametophyte is also haploid. So with this I came to the end of this class, I came to the end of this video. This was a part 1 of the sexual reproduction in flowering plants, where I discussed the events prior to pollination, and I will provide you the notes. Go through the notes till pollination, and after the pollination, the events after pollination, the pollination event as a whole, as well as the fertilization, the double fertilization, I will discuss in the part 2 of this chapter. So till then, take care and study well.